I'm from SciLife Lab, the National Genomics facility. So we are basically a core facility for genome sequencing in Sweden. And we have all types of instruments. We're located both in Stockholm and Uppsala. A lot of it is still done with short-read sequencing. But in Uppsala, we have been focusing for quite some time on using long-read sequencing. And this is for all types of applications. And we even take on projects from abroad. And when it comes to smart sequencing, we have quite a long history. So the first PacBio RS2 instrument was installed back in 2013. And then we were very happy last year to receive the PacBio Revio system in March. So we have been able to use this machine now for about a year. And I'll go through a bit sort of about the technical things that we have found with this instrument and sort of how it performs, and also a bit about the plans of what we're thinking to do next. So we got the machine in March 2023, and so far you can see that the number of smart cells here that we have run each week. So it's been a few gaps, a bit of up and down, but we were able to process 105 smart cells last year. Then we had a nice long Christmas break, and then we came back, and so far we have been sequencing 116 smart cells this year. So actually more sequencing this year than all of last year. And what are we then doing with this instrument? Well, we're running different types of applications. I've divided them into like 3 different categories here, and I will come back to these later. So we have a very big piece of this pie that is human medical sequencing. So it's 112 smart cells so far. And this is kind of interesting because this piece of the pie was like non-existing before on the Sequel 2 system. So it's becoming very popular for these types of applications. Then we have biodiversity, which is about 100 smart cells. This is something that we've been doing for quite some time. You can also see the different types of like species that we are sequencing. And then we have one group of samples here in the very bottom here that is not so much yet. RNA and single cell sequencing, only 6 smart cells so far. We're hoping to do much more. Also, I have here a slide showing the yield for the different sample types, because what we have seen is that different types of samples behave very differently on the Revio system. So now we can also sort of try to estimate the number of gigabytes that we can get from each of the sample types. But this will also depend very much on the tissue and so on that we're sequencing. But at least it could give us perhaps some guidance when we talk to our customers about how many smart cells they will likely be using for a project. And also I've included here, if you're interested, the total amount of hi-fi data we have generated so far on our Revio system is 15.4 terabytes or terabases. So it could be interesting for the competition, perhaps. Yes, and one thing that is very key for all these experiments, since we're running all different types of samples, is DNA extraction. I will not go into the details of how this is done, simply because I don't know myself. But we have experts here. So, for example, Julia Tooley, they're working with this. And they can sort of discuss more on which types of preps we're using for different types of samples, because it can make a huge difference. And I also discussed with our project coordinator, Sanna, about our standard Revio workflow, because I wanted to show sort of how we're doing this right now. I don't know if it's the optimal approach, but this is how we're doing. So we first shear the samples. We're using the Megaruptor 3 system with speed 29 to 31. And it's a bit random sort of how long fragments we get, but usually it's in a quite good range for hi-fi sequencing. The next important step is the size selection. So we're using right now the Pippin HT system. And we have a lower cutoff at 10 kbs, and there's a higher cutoff at 50 kbs. So we're trying to sort of get the fragments that could become good hi-fi reads in this way. And then for the actual run, we're using a 250 picomolar binding. We're doing adaptive loading and 30-hour movie time. yeah, so that's how we're doing. And I would also like to talk about some collaborations that we have, because we take on external projects, but we also have more sort of collaborations with institutes that are doing different types of studies. And in these three different areas, we are involved in biodiversity with the Biodiversity Genomics Europe and the ERGA projects. So it's my colleague Olga who knows everything about this. There's also an interesting project on RNA and single-cell sequencing. So Anna Conesa is here in the audience, who leads this Long Trek consortium, which is a very nice doctoral network that are trying to develop the new tools for studying this type of information, because I think that there's a lot to be done there. And also in the human medical sequencing, we're very interested in the rare disease, as we've heard about today. And Lars Föck will talk tomorrow about the things that we're doing in this area. So we are collaborating with Genomics Medicine Sweden, which is like a network of hospitals around the country, and also the Undiagnosed Disease Network. We're also involved in the Hi-Fi Solves. I didn't find your logo, so I didn't put it here, but it should be here too. So this is sort of what we have been doing, but I would like to now focus more about what we think that we will run next year. It's always difficult to know, so this is like maybe just a guess at this point. But I think that we will do more biodiversity. I mean, there are many more genomes to be sequenced, so I think that we will do more of that. For sure, we will be doing more of RNA and single-cell sequencing, because we have very low numbers now, and also with the Kinnex kits, it can become very, affordable to do that type of assay. And with the human medical, I think that we will do actually much more. So I said that I will not talk about the rare disease stuff, but I will talk more about the population genomics that we're thinking of when it comes to the human sequencing. So I was involved in a project quite some time ago now, where we generated like a reference genome for Sweden. So we sequenced 1,000 genomes with Illumina. And at this point in time, I thought it was kind of a boring project when it all started, but it became a very important resource actually. So it's used in the clinic and also for many different types of research projects. And maybe now it's about time to do the same thing also with long reads, because we can simply use the Illumina sequences are not suitable to use as references for the long read data. So I think that this is something that we really need to do, and we already sort of are thinking about the plans of actually doing this. And one thing here that we have compiled is like a wish list, also based on the previous experiences with the Sweden data. This whole project, we had perhaps at the top here, one issue that we need to solve. I mean, we need to have good consent for data sharing to make this as useful as possible. And also, of course, we need to find a source of samples where we can get good enough DNA and also the amounts that we need. But then there are also a few other things that could be important. Like if we have more phenotype information, we should also try to cover like a cross-section of the country. Additional omics data would be great, like proteomics, RNA, and so on. Available SNP array data is good, because then we can select the individuals, perhaps in a clever way. And of course, funding and resources will be needed to do this. So where are we right now? Actually, I cannot talk too much about the exact plans. I think it will become more clear in a couple of weeks. But we have been already sort of identifying a suitable sample cohort that we can use for this project. And right now, we're in the process here of obtaining the ethical permits and consent. So that's a very big thing, but I hope that it can be done quite soon. We also hope that we will get the funding and resources. It looks very promising. And then we will start towards the end of the year to run maybe 500 to 1,000 long-read whole genomes. That's sort of the initial aim, at least. And the last point here is that it would be very interesting to also collaborate with other countries that are thinking in similar ways to perhaps align these types of efforts. But, I mean, this is not so easy because there are a few challenges. Some of them were also discussed during the workshops earlier. So when scaling up, one thing is the automation. I mean, we need we need then to process many samples, so we need to do it in an efficient way. Can we use robots? I don't know, hopefully, but this is something we really need to think about now. The next thing is the analyses. But here I think we already have sort of a solution. So we have a bioinformatician, Felix, now, who has developed a pipeline, and we're using the Nextflow system because it's something, that the bioinformatics community is using quite a lot. So it could also be a collaborative effort here. And the kind of output that we get currently is like quality plots. We get the phased reads, we get the variants, SNVs, SVs, tandem repeats, copy number variants. It's all using the tools that were discussed earlier. We also, in fact, do a diploid assembly of all the genomes just because we can, and we map them back to the reference. Also, we get the methylation data. So I think this part, it's becoming quite solved. And then there's the last one, is about sort of if we can increase the throughput of the system. It's also been discussed a bit. And right now we're seeing like 110, 15 gigabases for our very best runs. And there's a lot of like disinformation on the Twitter X platform nowadays, but some people are claiming that they can reach 127 gigabases. But then it starts to become really interesting, because if we can reach like 120 gigabases throughput, then we could think about perhaps multiplexing 6 samples across 4 smart cells, and then get 80 gigs per sample, which would be great. But there are a few issues here as well. We need then to reliably obtain 120 gigs per smart cell. And perhaps the last part is even more difficult, to get like a near perfect barcode balance. So I guess that we'll see. Also, leading up to this conference, we made like an experiment. So I was interested in seeing like, could we just like increase the read length and then get more throughput? We are doing the 10 KB size selection, and then we can get like 100 gigabases of data. But if we go up to 15 KBs, would we then get like 150 gigabases? That was my hope, but it turned out to be not so successful. So that's perhaps not the easy solution. And one sort of interesting thing is that we only got 18.7 KB average read length for some reason. So in some cases, we get longer read lengths with the 10 KB size selection. But I guess it all depends also about on the sample source here. So there are many factors going into this. I also did like a plot just showing the hi-fi yield versus the average read length. And it's not so easy. You can't simply just increase the read length, it seems, to get more data. But it would be super interesting to discuss more about this. And just finally, these are the things that we're doing now, but are there things also missing from this chart, like things that could become important applications in the future? So we're trying to look into that as well and seeing what new and exciting things we can do. And here I've put up like 3 different things that we have been working on, or our colleagues at the other institutes are working on. So first, we have single-cell whole genome sequencing. Not sequencing the RNA, but sequencing the DNA in single cells. So we were able to generate a decent coverage of 1 human single cell using an approach with amplification. And I think this could be very important, especially when looking at mosaicism, perhaps in the brain and so on, which is difficult to study otherwise. So I think, and also people are quite excited about that kind of application. And also I want to mention spatial long-read RNA sequencing. So there's a group in Stockholm who is developing a very cool spatial VDJ application. So they sort of prepare the libraries and send them off to us, and they also published this very nice paper in Science describing their method. And finally, sort of one of my favorite projects is also on genome editing verification, because it seems like when we're using the CRISPR-Cas9 system for doing editing in cells, then we get structural variants both on the on-target and the off-target sites. And if you're thinking about using this for medical purposes, you want to have a good way of verifying this. And I think that you have to use long reads in that case. Yeah, so I think that was it. I also would like to mention that we have a conference coming up this autumn in Uppsala. And PacBio is a gold sponsor on this conference. You can submit abstracts, get a poster or a presentation in this very nice venue. Yes, so please check that out. And finally, I would like to end with a bit outdated picture of my colleagues. So we should take a new picture at some point, because I don't think everyone is in here. Yes, and thank you all for listening.